Welcome back, friends. In previous video discussions we have talked about the different types of eukaryotic transcription, and we have talked about the introduction, and we have talked about the role of RNA polymerase 1 and also the RNA polymerase 3 for the RNA transcription part. Now in this particular video we'll be talking about polymerase 2 and its role in the production of all the different mRNAs, which is coding for all the type of proteins inside our body in case of the eukaryotic system. Now in this case we'll be looking at the functionality of this RNA polymerase 2, which is the most important polymerase of all in case of eukaryotic system, and also how it initiates the transcription process in the very first point. Because the initiation, remember I've told you always that the initiation is varying from prokaryotes to eukaryotes, or even in case of eukaryotic different polymerase will have different types of initiation, but and also termination will vary, but we will see termination in totally different video. But in this video we'll be talking about the polymerase 2 and how it starts the RNA transcription in the very first place. And we will also see the role of general transcription factors, or GTFs, in case of this polymerase 2 mediated process. So let us consider this as polymerase 2 mediated process from here. So in polymerase 2, what we can have, again we will have the DNA. So three things are important. First thing is the DNA sequence, second thing is the polymerase itself, and third thing is the generalized transcription factors. And the interaction between the DNA sequence and the polymerase is the most crucial of all, as we can see in case of polymerase 1 and polymerase 3 mediated processes. So again in this case it is much more complicated than the polymerase 1 and polymerase 3. It will copy the whole DNA and produce the RNA according to the choice, and the elongation process again will remain the same, but it will vary in the initiation. Now we'll be seeing the initiation in detail and how the initiation is actually achieved. So let us consider. So for that we always have gone through these studies via a line that we will study the promoter region first, then we will study the pol and how the pol interacts with the promoter, because this interaction along with this generalized transcription factors are the key things for initiating any transcription process. Okay, so let us consider this. So the promoter region for this process is saying that we are having this DNA. So in case of this we are having two different regions actually among the promoter. Not actually, it is three different regions in the promoter for the polymerase 2 mediated process. One is the Tata box, which is very important, for the recognition of the polymerase to the DNA in the very first place. We can see this Tata box and also Pre-Transcription box in case of prokaryotic systems. Now second thing is the Initiator box. Initiator box is another important part, as the name suggests. From this box, or this box means these are not box like things, they are simply the stretch of nucleotide sequences inside the DNA. So they are stretch of nucleotide sequences. So again, initiator box means the stretch of nucleotide sequences from where the initiation of transcription begins. Okay, and third thing here, let me write it in the different color. It is the upstream elements, upstream controller elements, or upstream elements, whatever you can write it. Upstream elements are also an important part. Now again, like the other part, these upstream elements will bind with this RNA polymerase along with activators, along with mediators. Okay, and those mediators and those activators could be transcription factors, they could be specialized transcription factors. Okay, now this part will behave a little bit differently from that we can see in case of pol 1 and pol 3. This part, in the previous cases, it will bind to some transcription factors, then it will recruit an addressing molecule. Remember that addressing molecule will address the polymerase to the place of the initiation, then the melting of the complex, and start of the transcription. This is the basic view for Pol 1 and Pol 3 mediated transcriptions. But in case of Pol 2 mediated transcription, it is a little bit difficult. It is different. It is tightly controlled. And in this case, we will be seeing the upstream control elements having a slightly different function. It will not only recruit those transcription factors, but specialized transcription factors. Remember, we are having two different types of transcription factor. One is generalized transcription factors, one other one is specialized transcription factors. Generalized transcription factors are vigorously found, and we have seen the effect and the role of generalized transcription factors. Now in this case, for the first time, we are going to see the role of specialized transcription factors in the processing, in the production of mRNA, okay, inside the eukaryotic system. Now these three things are very important, and these are the construct of the promoter, typical promoter sequence. Okay, so I am telling this because this promoter sequence is nothing like a small region. It's a huge region. In case of prokaryotic system, remember when we used to draw prokaryotic DNA and prokaryotic promoter, we used to draw like this. We used to just put here like that, and we'd say that this is the promoter, this is the operator, and these are the coding genes, right? Like that. When you study operons, so just go back and study the operon concept, you will find something like that. Just small stretch of sequence is the promoter. But in case of eukaryotic system, promoter may not be placed only once. So what happens in this case most of the time, so we are going to see here also. So say this is the DNA of our interest. Now in this DNA of our interest, we are having the Tata box here. So this is the Tata box, and further downstream we will have the initiator box. A small downstream of that we are having the initiator box, and upstream of the Tata box, far upstream of the Tata box, what we can have this upstream element, UPS. I just write it like UPS. I don't think you may see this, so UPS. These are the upstream element. Okay, so these are the upstream elements. So these three things combined together is called a promoter for this Pol 2 mediated RNA synthesis. So this is something new, right? So in case of prokaryotes, we can find a small stretch, this is promoter, but here the promoter may be of say 200-300 base pairs long regions. In between there are small regions that are not required for this particular process, but or maybe they have some job but we haven't figured it out. It should be a possibility also, but still these parts are shown to have a remarkable effect on the transcription of mRNA from the DNA in case of eukaryotic system via the Pol 2. Okay, now we'll be seeing that the role of this process. So we are having this DNA, inside them we are having these sequences. These three things are important: Tata box, initiator box, and the upstream elements are, so let us USEs, actually UPS or USEs we can write. So let's write upstream element. It will be better to write it like USE, upstream elements. Okay, now there are general transcription factors involved in this process. There are specialized transcription factors involved in this process. Now the generalized transcription factor will interact with the Tata box as well as initiator box. Remember, GTFs will interact with Tata box and initiator box for this process, and the typical generalized transcription factors that are in use, remember how the nomenclature comes in, they will be called transcription factor 2, because we are talking about the transcription factors involved with Pol 2 or RNA Pol 2. That's why the nomenclature will be transcription factor 2. Now it could be transcription factor 2b, transcription factor 2a, transcription factor 2d, 2h, 2i, 2j, 2f, and so on like that. Okay, so these are the variation of the names, but the nomenclature will be transcription factor 2 in this case. They are among the generalized transcription factors. In case of specialized transcription factors, these STFs are going to bind with upstream elements of this region. This is important. This specialized transcription factor will bind with upstream element, as well as they are going to bind with the polymerase. Okay, so this specialized transcription factor, as the name suggests, they are specialized, they are specific, they are special type. So they will do some special job, and in this case the job is to make sure that the polymerase is correctly interacting with the DNA, as well as it produces the signal to the polymerase that you are in right place. Now you can go for the elongation step. So it will give a signal to the polymerase that yes, you are in the right place and please go and do the transcription. Okay, so it is important for binding of STFs in two different places. One is UACs, another one is the polymerase itself. So remember, UACs are further upstream, polymerase is further downstream. So these molecules must have some ability to bend the DNA to, to attach both the things together, right? What it means, it will be clear in the few minutes, so don't bother about that. So here until this point, up to this point, what we know is these 3 points: GTFs will bind these two regions, STF will bind this along with the polymerase. Now RNA polymerase is the most important player of all. Now before initiation, we know in any kind of initiation we must need proper interaction of these transcription factors, a proper interaction of polymerase with the DNA sequence, DNA promoter region. And for this proper interaction of polymerase with the DNA sequence, we need the presence of transcription factors. And here is the transcription factors, GTFs, or generalized transcription factors. So what it will do is that it will bring some generalized transcription factors onto the place. For example, in the very first place, this Tf2b Tf2d is come and going to bind with this Tata region. Now this Tf2d protein, so let me write it here, the Tf2d protein, so this is Tf2d. Now this Tf2d protein is really important protein. This protein is having many subunits, they combine together to make this large protein. Now this Tf2d protein, one simple part of this Tf2d protein is providing the higher affinity towards this Tata sequence inside the DNA, and that particular stretch of the 2F2D protein is called Tata binding protein or Tata box binding protein or TBP. It's a part, remember, it's a part of Tf2D protein which is called the Tata box binding protein. It is having higher affinity to the Tata box of this DNA sequence. Now as it bind with this Tata box region, it will make some conformational change in the DNA sequence, and this conformational change in turn bend the DNA minus 80 degree up to minus 80 degree. So it will cause the bending of DNA minus 80 degree. So this is the very first type of reaction that is going to happen inside this DNA along with the interaction between GTF and DNA Tata sequence. So the very first interaction of this process will be that Tf2d will come, and the TBP part of that is going to attach with this Tata region. It will bend the DNA minus 80 degree. Now as it bends 80 degree, remember 80 degree is a lot, 90 is just straight like that, 80 is slightly bended, so it will make a structure like that in the very first place. So say like that. Now here is our protein. This is Tf2d, or you can write it TBP region of the Tf2d, and here is, remember, our initiator, and here is our upstream elements, and this is our Tata region, right? This is our Tata region. So it bends the DNA like that. Now how this protein can bend this DNA? So I'm just going to tell a little bit about that. That this protein is consisting of this Tata binding protein or Tata box binding protein. Now this Tata box binding protein or Tata box binding factor, whatever we are calling it, this is providing beta strand like structures. The protein is made up with beta strands or beta sheets. Now these beta strands are going to interact with the minor groove of this DNA. So the beta strands are going to interact with the minor groove of the DNA. Now, as it is interacting with the minor groove, the beta strand is having lot of phenylalanine residues. Remember this, they are having a lot of phenylalanine residues, polyphenylalanine residues. So these polyphenylalanine residues that are present in this TBF, they are kicking the minor groups to come to be bended, and this kicking is very much important. So they kick it and make it a bend, and when it is bended, at that point this protein, the other regions of the protein, the other regions of that beta strand is going to form hydrogen bonds with that bending conformation. So first thing, polyphenylalanine residues kick it to be bended. Second thing, other regions of that protein is going to attach with that bended conformation via the hydrogen bonding. Now it stabilizes at this point. So what it happens, suppose you are having a flexible wire like that, you just bend it and hold it together in your hand like that, so it will never be straight again, right? So it bend it like that. This is the very first event of all. Now after this event has established, the regions of upstream element and initiator region, you can see they come really closer. So it is bringing upstream element and initiator region closer. Why? Because it is having a reason for that. Okay. Now second point is, in this case, then it will recruit other proteins. Right after the binding of Tf2d, it will recruit Tf2a. Tf2a will come and attach to it this Tf2d and the DNA to make this complex stable. So let us draw Tf2a. This is not that much important, so just let me draw it. Tf2a, it will come and bind with it to make this complex much more stable. Now it stabilizes the complex. Now the DNA is bended. After that, it will recruit Tf2b. Okay, now Tf2b protein is the first protein which is going to recognize the initiator box. So it will be recruited directly to the initiator box. So here is the Tf2b, Tf2b protein. Say this is Tf2b. It will be sitting on the initiator box, and then it just cover up everything. Just like Pol 1 and Pol 3 mediated process, we have seen the GTFs will start soon to cover the different regions of the DNA, and it is covering the different regions. Tf2b is coming and sitting onto the initiator box, and then everything is filled out. At this point, this Tf2b in this case, remember this Tf2b in this case is acting like that addressing molecule. It is sensing the environment, so no other regions are free. So it's a long stretch of DNA. I'm just at the beginning of the coding sequence. We haven't talked about coding sequence. Coding sequence will be further downstream. So at this point, whenever these things are bound with the DNA, it eventually provides the signal to RNA polymerase that it's a time to come and bind. Because whenever simple DNA is there only, polymerase II has not that much affinity to only single DNA or naked DNA. Whenever DNA is bound with these transcription factors, it increases the affinity of this DNA to the polymerase II. Then polymerase II can come and sit onto it and bind with it. So it gives a signal, and polymerase II along with one addressing molecule will come and attach to it. Now the addressing factor again in this case is Tf2f. So Tf2f in this case is the factor. So here comes the Pol 2. I always draw Pol 2 in the blue color. So say this is the Pol 2, and along with Pol 2 we are having say Tf2f. So this is the complex. This complex and this Pol 2 obviously is having a hand. What is this hand? We will talk later. Now it will bring this Pol 2 and attach it to this complex. Okay, so let us draw it. It will become really very difficult for you to visualize. So say this is the polymerase. Polymerase has bound with it, and along with the polymerase, Tf2f is also bound with this, and this is the situation. And here is the hand of Pol2. I'm just drawing this hand like that. It's a hand of Pol2. Why it is important, we'll be seeing it later. So this happens. So Tf2f will bring Pol2 and make it sit onto this initiator complex or the transcription complex. It is called a pre-transcription complex. Pre-transcription means before transcription. So it's a transcription is not started yet. So this is the very beginning of the transcription. That's why the complex which is at the beginning of the transcription is called the pre-transcription complex. Now in this pre-transcription complex, what we are having Tf2d, Tf2b, Tf2a, then Tf2f, and it will recruiting the polymerase II. Now here comes the important part and the role of STF or specialized transcription factors. Now specialized transcription factors, remember, they can interact with upstream element as well as the RNA polymerase. So now these specialized transcription factors will come and they will sit onto this upstream control element or upstream control elements. Now this upstream control element will be at so say this is the specialized factors. This is the interaction. So this is the specialized transcription factor. It will interact upstream control element and polymerase II both at the same time. So it is having two different domains for the binding. One is the DNA binding domain. Remember, this region is the DNA binding domain. With the help of this DNA binding domain, it is interacting with the upstream control element. And with, on the other hand, the RNA polymerase binding domain, with the help of this RNA polymerase binding domain, it is binding with the RNA polymerase II in this case. So this is specialized transcription factor. So this is DNA binding domain and this is polymerase binding domain. Okay, so with the help of two different domains, it is sitting and interacting on both the case. But it can only interact, or the interaction is possible only in those cases when the DNA got bended. That's why it is required for bending the DNA. And when these specialized transcription factors sitting onto this region and connecting these two things, connecting the polymerase and upstream control element, it will further bend the DNA and eventually it will tell the polymerase, now it's your right place, you have sit in the right place, now it's time to go, it's time to melt down the complex and initiate the transcription. It provides the signal, it is telling the polymerase that now you are sitting in the correct place, now just begin the process. Now as it senses and provides the signal, it will recruit some other generalized transcription factors, and in this case that generalized transcription factor is Tf2h. Now everything is correct, polymerase is sitting onto the place, it is sensing the signal and ST interacting, it is giving the signal here, this is the time. So the first event in this case will be melting down the DNA, then the incorporation of nucleotides, then the polymerization, right? So we have to melt the DNA now and go through the process. Now this is the signal STF is providing, STE is provide, STF, it will be STF anyways, STF is providing, and then it will recruit the protein which is responsible for the melting of the double-stranded DNA. And the protein which is helping in this melting is Tf2h. Okay, this Tf2h protein is very, very important protein. Why, we are going to see in a moment. So until this point, up to this point, I hope everything is clear. Now from that the signal comes, Tf2h will be recruited to this position, and this Tf2h will provide this RNA polymerase the signal. Now it's your time to work. Start working. And how this polymerase is going to get the signal, because there is nothing signal transduction like that, so no signal transduction molecules is there. So how this polymerase is going to get the signal? Here comes the importance of this arm of this RNA polymerase that I have drawn. This arm is, remember, RNA polymerase is having alpha terminal and C terminal domain, obviously. So C terminal domain is called the alpha CTD of this polymerase. So that alpha C terminal domain of this polymerase is the arm of this polymerase. Now this is a long arm, it's a long region of protein. That region can be modified, right? So simple modification of that arm, modification like phosphorylation, simple phosphorylation of this arm will tell this polymerase that it's a perfect time to initiate the process. And this program is also conducted by Tf2h. So you can see Tf2h is functioning in multiple ways. It is telling the RNA polymerase II to start the proper functioning, and it is also helping to melt the double strand structure of the DNA and initiate the process. Okay, so now we don't have area in this place, so let us come into this side and discuss it, because I don't want to rub this picture. It will be required for our summarization, so I am just okay. I hope this is a perfect region to draw this picture. Now here, this is the complex. Now at this point what will happen, they will bring this Tf2h. Now as the Tf2h will come, it will tell this, the Th2f functions in three different ways. First thing, it will phosphorylate the alpha CTD of this polymerase. Okay. Second thing, as it is phosphorylating, it will tell the polymerase it's a time to move on. So it's a time to move on. So just don't be seated at this point. Just start to move on. And as well as it will melt the DNA, because if it won't melt the DNA, polymerase will not be able to move on, right? So it is giving two signals. One is to the polymerase that it's time to move on, start moving on, and it is melting the DNA to clear the path for moving off the polymerase II. Okay, and the melting process here, remember the melting of DNA in case of the replication we have studied, it is conducted by the DNA helicase. So it's a hexameric protein which utilizes the energy from ATP hydrolysis to separate the two strands apart. Now in this case also, this part of the reaction required the ATP hydrolysis energy, and again the energy is utilized by the Tf2h protein, and it will help to separate two strands apart to produce what is called the open complex. So in this case, let me draw the structure here. Say this is our system, and I'm going to draw this polymerase only because we don't require other things, because once Tf2h comes in the place, so let us draw the Tf2h. Say this is the Tf2h. As the Tf2h come to the place, it is phosphorylating. Not only phosphorylating, but before phosphorylation of the CTD region, it will bring the CTD region outside. So the CTD region will change its position. In the previous place, it is just folded, and now it is open. So it is folded, now it's totally open, and it is ready for the phosphorylation. So it starts phosphorylating this region, and after it establishes this mission, all the other generalized transcription factors will go away, and only Tf2h will be attached to this RNA polymerase, and it will carry the reaction, because from there on they won't require any of these other generalized transcription factors, because the recognition part of the process, because in any transcription process the first part is the recognition, then the start or initiation. The recognition process is done where we require a lot of different generalized transcription factors as well as some specialized transcription factors, but in this case we won't need those factors again. Tf2h itself can do this factor to this thing along with the RNA polymerase. So polymerase will move on to this downstream region, so they will move on there and it will start melting the DNAs too. So let me draw the structure of melting of the DNA. So it will be something like that, say. So DNA again start to melt and polymerase, RNA polymerase is start to move onto this direction. Okay, and the process will start and the bending will be removed because all the TBP and factors are getting removed, getting lost. So the bending won't be there, bending is not required anymore because the reason for the bending is to good interaction between the upstream control element with the polymerase. So we won't require bending anymore, so bending will be gone and then polymerase will sit onto and will start moving forward. As it is moving forward, this Tf2h will drag energy from ATP hydrolysis and help this polymerase to move in the forward direction. Nucleotides will come in and attach to this complementary nucleotides that are presented by one of these template strands, and then polymerization will occur in the elongation stage of the process. So we can see how complicated this process in this case of eukaryotic system, in case of polymerase II mediated mRNA synthesis. Okay, now as it is producing the mRNA, at the end of the point we need to modify our mRNA with different types of modifications. For example, the modifications like polyadenylation at the 3' end and 5' capping. We won't bother about 5' capping because suppose this is the mRNA that is being produced. This is the 5' and this is towards this direction is the 3'. So 5', as soon as the 20 to 30 stretch of nucleotides just come out, 5' is just made, it will start making the 5' capping at this point. But at the 3' we have to concern because we can only have 3' only after the ending of the transcription. Then you have to grab it and make the polyadenylation, make 100 to 200 sequences of adenine attached to this 3' end. Okay, now at this point, this is the situation we have already discussed. So if you want to know detail about the 5' capping and 3' polyadenylation, just go back to my website, go back to my YouTube channel and search the video. You will find the 5' capping and 3' adenylation videos in detail. So just go them and come back. So I'm not going to talk about this again in detail. I'll be talking about the termination, but that will be not that much important now, because this termination means this polyadenylation process and how it terminates. So the termination in case of eukaryotic system is never achieved utilizing the row dependent and row independent manner. Instead, it is achieved via signal dependent cleavage. Okay, so when it gets some signal, it will start cleaving the growing RNA, and it will be adding the adenines at the terminal point. Okay, and the signal in this case, suppose after transcribing, the signal in this case is something like that. The signal is a AAU, AAA. This is the signal. Now as they reaches this type of signal, at that point they will recruit some factors again, recruit some proteins. That proteins is going to cleave the DNA from that point. So here the cleaving site is CA, so it will cleave just right after the CA, 5' C, 3'. So this is the sequence of CA. It will just cleave it after that, and then a protein called polyadenylation binding protein and also the polymerase will keep on adding adenines onto the last 3' region of it. So they will keep on adding adenines there like that. After adding the adenines, poly A binding protein will come and sit onto the region and stabilizes the structure. And meanwhile, when suppose during this process, say here is the signal, so it cut this, so this part of the RNA will come out like that, right? So try to understand this concept. This part, there is a signal, it cut it from this, the endonucleus cut it from here. This part of the mRNA now is in our hand, and it is again polyadenylated, and polyadenylation binding protein sit onto that. Everything is happened, but the polymerase keep on transcribing, though it cut it from the upstream region, but still it will keep on transcribing, whether that stage of the RNA is not at all required for us, but it will keep on transcribing. For example, suppose it will keep on transcribing from here to up to this point, so we'll get this stretch of sequences later. This stretch of sequence is not required for us, we know that, because it is not having the coding regions. It's just the spontaneous movement of the polymerase, because as soon as it reaches the signal and cleave it, polymerase won't stop, and all the transcription machinery won't stop. It will stop after some time. During that time, it will synthesize a short stretch of RNA, which is not at all required for any purpose. Now, this type of RNAs will be thoroughly degraded inside the nucleus, even after moving into the cytoplasm. So, this sequence will not be allowed to come to the cytoplasm. It will be chewed out, degraded inside the nucleus as it is making. Okay, so here it is a type of ending, type of termination. So I'm not going to go into detail about the termination and polyadenylation thing. If you want to learn, just go back and see my video on polyadenylation. Now let us summarize whole process. So from the beginning, what we've got this sequence, and the promoter means Tata box, initiator box, Tata box in the middle, initiator box downstream of the Tata box, and the upstream control element is further upstream of the Tata box. Now here, in the very beginning, Tf2d will bind along with this Tata binding factor, Tata binding protein activity, and also some other factors called Tata binding factors or TBFs. Now here it will recruit Tf2a. It will bend the DNA, and for the stronger stabilization of the bending state of the DNA, it will recruit Tf2a. Now the bend DNA is there, then it will recruit Tf2b onto the initiator site. When it binds to the Tf2b binds to the initiator site, it creates a higher affinity state of the DNA protein complex that will recruit Tf2f and polymerase II. Now Tf2f and polymerase II is a very important complex. Now Tf2f is the addressing molecule in this sense, which will bring the polymerase II and keep it on binding to this bended form of the DNA. And then after binding of this polymerase, specialized transcription factors will be recruited, which will bind with upstream control element as well as the RNA polymerase at the same time with these two domains. One is the DNA binding domain, another one is the polymerase binding domain. After this binding, it will provide the signal to the polymerase and will recruit Tf2H. Tf2H is having multifunctionality. This Tf2H will phosphorylate the C-terminal domain and will make the C-terminal domain open, and then phosphorylate the C-terminal domain. This provides the signal to the polymerase to move on forward direction, and forward means 5' to 3' direction here. Let me write it here. Forward means 5' to 3' direction. Sorry. Okay, and at the same time it is providing it is taking the energy from ATP hydrolysis and melting the DNA. So it melts the DNA and then start addition of nucleotides, polymerization, and the movement of polymerase at the 5' to 3' direction. It will start elongation process, and that's how at the end termination happens and mRNA is produced. So this is a process of mRNA production utilizing the RNA polymerase II inside the eukaryotic system. This is a complicated process. Go through several times. I hope this video will help you. Thank you.